Please welcome to the stage Concord, North Carolina, Team 2, Mission 11 Flight Experiment.
Good afternoon. We are from Concord, North Carolina. I'm Paige.
I'm Zainab.
And I'm Shri.
Our experiment is about the gravitropism in radish seedlings and microgravity. Our teacher facilitator was Ms. McNutt, and we are from Concord, North Carolina. The goal of our experiment is to find the differences between the development of radish seeds and gravity and microgravity. We also hope to learn about how gravitropism differs in these two environments with varying degrees of gravitational force.
Our experiment will contribute to the start of agriculture in space. It is important that a plant develops properly in its early stages so that it can eventually serve its purpose for food, medicinal properties, or the oxygen cycle. If plants are not able to grow in space, that means that these things will have to be provided from Earth, which would be expensive.
In our experiment, we will be trying to understand how gravitropism varies between gravity and microgravity. Gravitropism is a plant's response to the stimuli of gravity. When amyloplasts, a type of organelle, settle to the bottom of a plant cell due to gravity, they touch the endoplasmic reticulum of the cell. The endoplasmic reticulum, in response, releases calcium ions. The calcium ions trigger the release of indole acetic acid, a type of auxin. In the roots, this slows the growth in the bottom side, causing them to curve. The roots grow along with gravity, which is positive gravitropism. In the shoot, indole acetic acid stimulates growth, which allows it to grow against gravity. No matter what position the plant is in, the shoot will always grow straight and against gravity. This is negative gravitropism. In our ground results, our seedlings in our FME mini labs range from being 1 centimeter to 3 centimeters. Most of the seedlings were made of the primary root and the rest of the cotyledons. The roots had begun to curve while the stems were relatively straight. However, we predict that the opposite will happen in microgravity. We hypothesized the shoots of the seedlings grown aboard the ISS will be larger and the roots smaller than the seedlings grown on Earth. This is because the shoots will have less gravity to grow against and the roots will have less to grow along with. Negative gravitropism will be stronger. We also believe that the seedlings will not grow straight, but curve in random directions since gravitropism plays a major role in the direction of plant growth.
We are using the Type 3 FME Mini Lab 2, which has 3 compartments. Volume 1 will have 3.4 milliliters of sterilized water, which will activate our experiment, since it is one of the simplest ways to germinate a seed. Volume 2 will have 2.5 milliliters of sterilized soil and 3 radish seeds, or Raphanus sativus seeds. We chose these seeds because they are easy to grow and maintain, which will help since we cannot tend to them. We originally planned to tuck the seeds into paper towels, but they grow better in soil. Volume 3 will contain 2.5 milliliters of formalin to preserve our experiment.
On day AO, clamp A will be unclamped, then the mini lab will be shaken vigorously for 20 seconds. This will release water into the soil compartment, activating germination. The same will be done for clamp B on day U14, releasing formalin into the mixture, preserving the specimens.
Coming August, our experiment will be launched and sent to the ISS as part of Mission 11. When it comes back, and if our results match our hypothesis, this means that our idea of gravitropism in microgravity is right. There will not be enough gravity in space for gravitropism to occur in plants, so the shoot will have an advantage and grow upwards, while the roots may slightly struggle. Different parts of the plant may grow in different directions, without indole acetic acid to control them. If our experiment shows otherwise, that will become an opportunity for us to do more research and understand why that was our result in microgravity. The research we have done throughout this experiment has taught us a lot about germination, plants in space, and gravitropism. We hope that our results will teach us more and expand the knowledge we have of plants in space.
We would like to take We would like to take the time to thank these partners institutions for making our flight experiment possible. Thank you to J. Ann Fries Magnet Middle School, with special thanks to our teacher facilitator, Megan McNutt. Thank you to the Cabarrus County STEM Program, the Borough's Welcome Fund, National North Carolina Space Grant Consortium, the for the Advancement of Science and Space, Subaru of America Incorporated, the Smithsonian National Air and Space Museum, and all parents, teachers, and community support we have received.
Thank you.